Hello, everyone. Welcome. Thank you for joining us for today's webinar. My name is Joy, and I'm a research scientist at Creative Biolabs. Today, we are presenting ConvertibleCAR-T, a new highly adaptable CAR-T platform to fight HIV, presented by Dr. Eytan Herzig. Just a little housekeeping before we get started. At this time, all participants are on listen-only mode. If you have any questions during the presentation, please type them into the question box in your Q&A panel. We will have time for questions at the end. The Q&A panel is on the right-hand side of your screen. Now I'd like to introduce today's presenter, Dr. Eytan Herzig. Dr. Eytan did his PhD at the Tel Aviv University and worked as a postdoctoral fellow at Tel Aviv University and Gladstone Institute of Virology and Immunology of UCSF. Now he's a research staff scientist in the Gladstone Center for HIV Cure Research, where he leads the use of new technologies to fight HIV. Now I would like to turn the meeting over to Eytan. Thank you. You may begin.
Thank you, Joy, for inviting me to give this webinar. Thank you, Biolabs, Creative Biolabs. And today I'm going to talk about the convertible CAR-T, and it is a highly adaptable CAR-T platform to fight HIV. I'm going to start with a short introduction about HIV and the latent reservoir. After that, I'm going to explain a little bit about our approaches to achieve HIV functional cure, introduce the new CAR-T platform called ConvertibleCAR-T, show some advantages of those convertible CAR-T. I'm going to show some results that we obtained with the convertible CAR-T and the way convertible CAR-T kills HIV-infected cells, some advantages and practical issues that we still have with that platform, and then I'm going to summarize what I've showed you today. So we're going to start with introduction to HIV, human immunodeficiency virus. It's an envelope RNA virus. It's a retrovirus. Therefore, it has a high mutation rate. The modes of transmission of HIV are sexual contact, blood transfusion, and IV drug use, and from mother to child. The main target of HIV is CD4 T cells. There's CD4 depletion leads to acquired immunodeficiency syndrome, or AIDS. And even though we have antiretroviral therapy, we have a lot of drugs against HIV, we call them ART, there is still no cure. And when you look at the HIV epidemic until today, you can see that more than 30 million people have died from HIV/AIDS related death. You can see on the top row that still more than 37 million people are living with HIV today. Less than 1 million are going to die every year from HIV-related death, and amongst them, around 100,000 kids under the age of 15. When you look at HIV and how the life cycle of HIV goes, you see that in the beginning there is a lot of circulating viruses in the blood until this HIV-infected individual is getting ART, or antiretroviral therapy. When the patient gets those drugs, the circulating viruses goes to less than the detectable level, and then as long as this person is on ART, we do not see viruses go around. But what happens if we stop ART for different reasons? Some people cannot adhere to this treatment. The virus is rebounding really, really fast, as short as a week or two. And the question is why the virus comes back, even though we had this patient on ART for months or even years. In order to understand that, we need to look at the HIV life cycle. This is an oversimplified model of HIV life cycle. The virus starts with binding to the cell through the CD4 receptor and the CCR5 or CCR4 co-receptor. Then it fuses into the host membrane. It gets his genome, which is an RNA genome, into the cytosol, and then reverse transcribe it to a double-stranded DNA in step number 3. Step number 4 is in the integration. Those double-stranded DNA integrate into the host genome, and then it stays forever. It doesn't go outside. Later on, the virus uses the cell machinery in order to produce new genomes of the virus and new proteins. It assembles next to the membrane and then buds out. After the budding, there is a step called maturation. And after this maturation step, this virus is infective. It can start infecting new cells. And when I said that we have different HIV drugs, we have a lot of different drugs that can stop the binding of the virus, the fusion of the virus, the reverse transcriptase of the virus, and even the integration of the virus. And then we even have drugs that stop the maturation step of the virus. One thing that we cannot stop is taking the virus out of the cell. And part of this HIV life cycle is what we call latent or latency, when the virus at some point, either by itself or something that is imposed by the cell, decide to stop producing new viruses. So we have a virus inside the genome, but it doesn't produce any new genomes and any new proteins. So those cells called latent reservoir. And those cells are the cells that at some point, when we stop the ART, are going to start the new infection again. And what do we know about this latent reservoir? We know that it's established early after infection. It can happen in a couple of hours or a couple of days after infection. Most of the resting reservoir is resting memory CD4 T cells, but other cells have been applied to be involved, like microglia in the brain. Approximately 1 in any million CD4 T cell is latent, so it's a really rare event. And the maintenance of the reservoir can happen in two ways, either by those cells just dividing, so this is called clonal expansion, or in sanctuary places where ART do not get full penetration, then there is ongoing replication. And the most important thing about the latent reservoir is that this is the barrier for HIV cure. We do not manage to get an HIV cure because of that. So we ask ourselves, can we do something with this latent reservoir? And are there any natural examples for people who control the latent reservoir? And happily, we have two groups of people that can control the virus by themselves. Those are called elite controllers or post-treatment controllers. I'm going to talk a little bit about each of those groups. So the first group, elite controllers, those are a rare group of people that were infected with HIV, but they managed to keep low or undetectable levels of the virus without any treatment of antiretroviral therapy. Roughly 50% is linked to a genetic background. They have an increased CTL activity, but the problem is that they have higher inflammation, so they are having 77% higher hospitalization rates than people on ART. That's why nowadays people that are elite controllers are being given ART to control their inflammation. So this is a great group that we can learn from, but it's hard to imitate the genetic background. So we can go to the next group, which are post-treatment controllers, and as you can understand by the name, they were treated with ART. They usually started treatment very soon after infection, and when ART was discontinued from different reasons, no viral rebound was observed, although we have low levels of viral persistence. There is not a link to a genetic background, which is something that is good for us that we can imitate, and the nature of immunity is still unclear. It might involve killer cells called NK cells, and what we can see in those groups is that in this post-treatment controllers, they have a relatively low amount of reservoir and they have low inflammation. So we are thinking, what can we do in order to imitate those patients that are off ART and they're still able to control the latent reservoir? So if we wanted to reduce the reservoir, like in those post-treatment controllers, there are different or multiple approaches for that. It starts from reactivating the cells and then having the immune system kill those specific cells. We can send antibodies to those areas. You can send chimeric antigen receptor, as you can see here on the bottom. There's even option to silence those reservoir cells by either silencing the HIV-specific genome, or even use CRISPR-Cas9 to mutate or take parts of the virus outside. So what I'm going to show you today is a combination of three approaches that we can see here: the latency reversing agents to reactivate the cell, and then the use of antibodies and chimeric antigen receptors. And when we look at this system, we call it this approach called reduce and control, which is a strategy to recapitulate post-treatment controller by reducing the size of the reservoir and then let the immune system control the small reservoir without having this patient take out for the rest of their life. And this can be done by different immune modulator or even silencing of the small reservoir after we reduce the size of the reservoir. So when we look at reducing the reservoir, the question is how can we kill HIV-infected cells, or how can we kill latent cells? As you can see on the right is a CD8 CTL cell, it's a killer cells. On the left, it's HIV-infected cell, it's a latent cell. It doesn't express any viral protein, so it's basically invisible to the immune system. And when we use LRAs or latency reversing agents, those are small molecules that can reactivate those infected cells, we start to see those cells, those latent cells, expressing HIV envelope or HIV ANF on the surface. Now they're visible to the immune system and they can still shed some viruses, but those viruses can be stopped by antiretroviral therapy. So the question now is, can we use the immune system to recognize those HIV-infected cells? And unfortunately, the answer for that is no, for two reasons. One, HIV infection, there is a general cellular exhaustion. Those killer cells are exhausted as well, so they would not kill HIV-infected cells. And if the treatment or ART was given not in the acute phase, but even after a couple of months after the infection, there's already viruses that develop mutation against those killer cells. So we ask ourselves, can we create better killer cells? Can we introduce new killer cells that would substitute for the weakened immune system? And we thought, what about chimeric antigen receptor T cells or CAR-T cells? So what are CAR-T cells? CAR-T cells are T cells that being transduced by a viral vector to express a specific receptor on their surface. This chimeric antigen receptor can find different antigens on tumor cells or on target cells, and then signal through those signaling domains that you can see here for the CAR-T to kill by apoptosis the tumor cell. This is something that has been around for the past couple of decades, but only a couple of years ago it was really come to light when there are two FDA-approved treatment for B-cell malignancies against CD19. CD19 is expressed on B cells. So when we have B-cell malignancy in a person, we can extract white blood cells or T cells, and then transduce them to express specifically the anti-CD19 CAR. And after further expansion of those cells, we can introduce them back to the patient, and those CAR-T cells are now going to go around the body and kill those cells that are expressing CD19. So this is something that works really nice in CD19 malignancy. And when we look at the CAR-T by itself, we can see that there are some shortcomings, some problems that we have with the pioneer CAR-T approach, and we wanted to see how can we adapt it to fight HIV. So, for instance, CAR-T is only attacking single antigen targeting. And in HIV, since there's a lot of mutation, a lot of strains, this is something that we want to expand. Once the CAR-T cell is being transduced, it's always on. There's not a lot of control. This can lead to a lot of toxicity. And most of the CAR-T cells are mouse-derived single-chain Fc receptors. They're only part of the antibodies used, and those are based on mouse, so they might be more immunogenic. So we collaborated with Cyphos, a company that developed this platform. We thought, how can we improve that specific CAR-T? And what they thought was to try to disconnect those two parts. The effector cell and the targeting part would be on two different places, and then we can achieve better killing or better safety. So once you divide something into two, you need to reconnect them in some way, and they took advantage of the NKG2D pathway. It's a receptor and ligand that is being used to kill stressed cells by CTLs or by NK cells. And what they've done was taking the receptor, NKG2D, and the alpha-1, alpha-2 domain of the MYC ligand, and they made it an exclusive receptor and ligand system. So they just mutated the alpha-1, alpha-2, and they mutated the NKG2D. As you can see here on the bottom, the binding of this alpha-1, alpha-2 to the wild-type NKG2D is much lower than, the affinity is much lower than the binding to the mutant NKG2D, which gives us an exclusive receptor and ligand, and now we can bind to this specific mutant alpha-1, alpha-2, anything that we want. So for instance, if we want to make a CAR-T, we can bind to this alpha-1, alpha-2, full-size antibody. We call it MicAbody because it's the mic part and an antibody. We can bind even different immune modulators to those alpha-1, alpha-2, for instance, different cytokines, and I'll show you some results later on. We can even, if we want to do imaging, in vivo imaging, we can bind the fluorophore, and in the case that those cells become toxic, we can even deliver a signal to do CAR ablation. So when we look at those convertible CAR-T, we have now more than one antibody that we can use, which is really good. So when you combine it, you compare it to the conventional CAR-T that use only single chain FV CAR-T, you can see that now we can use multi-antigen targeting, not just one antibody. We can use different antibodies. It's not always on. It's off unless the MicAbody is introduced. So we can do MicAbody dosing, and it's not depend on mouse-derived single-chain V. It's humanized, human-derived NKG2D, which is supposed to be less immunogenic. So once we looked at that, we asked ourselves, we divided our system into two, but there is already a system, as you can see here on the left, the conventional CAR-T that works in the clinic as well. So we wanted to see if our system works at least as good as the FDA-approved one. What we did was tested it in one of the most recognized models in cancer. It's a lymphoma model where you inject Raji B cells or cancer cells. In our case, they're going to be fused to a reporter, a luciferase. As you can see here, what we did was introduce those specific Raji cells into the mice. And then after a while, we introduced those CAR-T cells, either the normal CAR-T cell or convertible CAR-T cell. And we gave by IP different MicAbodies. And in this case, our MicAbody is based on rituximab, which is anti-CD20, which binds to those specific Raji cells. What we see on the left is when you look at those mice, the control, after 21 days, they succumb to the tumor burden. But when you add the untransduced cell, those CAR-T cell parental cells that were not transduced to express the CAR-T, or the CAR-T by themselves without any antibody, you would see that we did not rescue those mice. But when you look here on the right, when you compare the standard CAR-T, the single chain FV, or the convertible CAR-T that we have with the Raji-specific antibodies, we managed to rescue all the animals. And this is something that we did killing in vivo, but we wanted to see, can we rejuvenate those cells in vivo? So as I told you, we can use MIC adapters. We can send immune modulators to those specific cells. In that case, we used a mutant IL-2, which has a lower affinity to the IL-2 receptor alpha, which means that it would not recognize, it would not bind to normal cells. It would only be in higher concentration next to convertible CAR-T cells. We did 3 doses, or 1 every week, after we induced the normal expansion and reduction in CAR-T. And as you can see here on the bottom, in black, you can see what happened when you don't give the immune modulators. There is an expansion after a couple of days after the injection. This is called the phase alpha. And then there is a contraction of the cells, both with the MIC adapter and not. And if you see here on the red triangles on the bottom, those are the days then we gave the MIC adapters. And as you can see, there is higher amount or higher concentration percentage of those convertible CAR-T in the blood. We got here almost 100%, but this can be modulated with the different IL-2 concentration. So that shows us that we can rejuvenate those convertible CAR-T in vivo, which is something that is really important to HIV that needs to be given repeatedly. You cannot do only one treatment, and I'll show you in the future, in the next couple of slides, why. So when we go back to HIV, we thought, okay, we have this convertible CAR-T. It works really nice in vivo for cancer, but what about HIV? What is the antibody that we're going to use? Which HIV antibody we're going to use? And as I told you, there are different strains. There are more than 200 different HIV strains. We cannot use one antibody. But what antibody are we going to use? And luckily for us, there are something called broadly neutralizing antibodies in rare HIV-infected individuals. 2 to 4 years after infection, they develop a broadly serologic neutralization activity with those specific antibodies. So those antibodies are called bNAbs, broadly neutralizing antibodies. They have a wide range of target. They can target between 10 to 200 different strains, but they have low affinity towards each of those strains. As you can see to the right, this is the structure of HIV-ANV. And what we did was took two different sets of bNAbs. We took bNAb that binds to one of the pockets, that is the CD4 binding site, and two antibodies that binds to the V3 glycan. And those are the MicAbodies or the antibodies that we decided to go forward. And the question is, can we use those antibodies in vivo? Are they going to make a difference? And as you can see here, those are on the top, there are two papers that shows that 3BNC117, an antibody that we are using, or MicAbody that we are using, can reduce the size of the infection in people, and it can suppress viral rebound for a few months. And that was recapitulated with other antibody, 1074, that is a V3 glycan antibody. And as you can see, it can suppress viremia. And on the bottom, you can see there is an ongoing clinical trial right now that try to combine those two different antibodies. So we know that it can work in vivo. Now we can go on and do our experiment with those ConvertibleCAR-T and those bNAb-based MicAbodies. So we can use more than one antibody. This is great. And now we ask ourselves, what is the best way to evaluate ConvertibleCAR-T for HIV cure? So the easiest way is to do it on cell lines or to take blood, but this is not where the HIV is hiding. So HIV hides in lymphoid tissues. So this is where we need to do our experiment, and we thought about that, and this is what we did with those findings. We tried to take fresh human tonsils and fresh human spleen of healthy donors, process them into what we call human lymphocyte aggregate culture, or HLAC, and then we infect those cells with HIV that is tagged with a GFP. We did it in order for us to monitor those GFP positive cells, or the green cells, and then we combined that with a ConvertibleCAR-T. We combined it with ConvertibleCAR-T and different MicAbodies for 48 hours, and later on we did FACS analysis, and we looked for two different cell types. One of those was the HIV-infected GFP positive cells, just to see if the ConvertibleCAR-T really kills HIV-infected cells. And even more important is what's happening when we do those experiments to the uninfected cells. Because if our system is going to kill HIV-infected cells and uninfected cells, this is not a safe system, and we would not want to proceed. So when we look back on that, we used different viruses, different antibodies, and different cells. We started with a couple of different viruses. One is BAL, which is a lab-adopted virus that binds to CD4 and the core receptor CCR5. And the other one that I'm going to show you today is called transmitted founder virus. This is a virus that can jump between people, so it can be really being transmitted. We use two different types of MicAbodies. I said 3BNC60 and 3BNC117 that binds to the CD4 binding site on HIV envelope. They're going to be in blue for the rest of my talk. And two that bind to the V3 loop, PGT121 and 1074. They're going to be in orange. And in order to control for those MicAbodies, we use two different negative control for our purposes MicAbodies. One that binds to B cells, anti-CD20, based on rituximab, and one that binds to HER2 on HER2-producing cancer cells. In order to control for our cells, we took the parental cells, those CD8 or T cells that we use to transduce to make CAR-T. We took them as a control for our CAR-T cells. When we go to start our experiment, we ask ourselves, what is the effector to target ratio that we have to do in order to kill those HIV-infected cells? Because we want to give the minimal amount of effector cells to not introduce any toxicity. When we looked and we did some effector to target ratio experiment, you can see here from left to right, the more effector cells you bring in, the more reduction of HIV-infected cells in green, as you can see here. Y-axis is the relative CD4 viability. It starts with 1, and then it goes down as we add more and more ConvertibleCAR-T. But the most important thing here is to see the toxicity. As you can see here in gray, we got those experiment with the toxicity for the uninfected cells, what we saw is that when we get to a 20:1 effector to target ratio, we do see some reduction in gray, which means that we do kill some uninfected cells. So we decided to move on, and all our experiments from now on are going to be in effector to target ratio of 10. So when we don't kill uninfected cells, we kill only infected cells. Then we moved on to kill HIV infected cells from CD4 T cells, from tonsils, from lymphoid tissue, and with our CCR5 tropic HIV. And what we saw is when we look at the controls, the green and the gray are going to be the GFP positive and GFP negative, or HIV infected cells and uninfected cells. As you can see here with all our 4 controls, either no CAR-T or just CAR-T with no antibody, or in the 2 right panels, you can see those negative control MicAbodies. We do not kill HIV infected or uninfected cells. And then the question is, what happens if we add just the MicAbody that is targeting HIV? As then you can see here, no control, no CAR-T, but just one of the HIV-specific MicAbodies. We do not kill HIV infected or uninfected cells. When you add the CAR-T to this HIV MicAbody, you can see that from 0.1 to 10 nanomolar of MicAbodies, you get around 60% killing of HIV infected cells, but you do not kill any of the uninfected cells. And this is something that we saw with the 3BNC60, and the 3BNC117 showed the same result. And the other 2 MicAbodies that are V3 loop dependent, they are showing the same results. So this is something that we saw here, and this is great. This is a lab-adopted strain, and we saw that all 4 antibodies can recognize and kill those HIV-infected cells. But what we saw is that there is no dose response. So regardless if we put 1.1 or 10 nanomolar, we see the same result, and there is no complete killing. So we thought, okay, how can we improve it? And we moved on to do some experiment with the founder strain, the transmitted founder strain, which is more clinically relevant. And we thought, if we reduce the MicAbody concentration from the nanomolar concentration to the picomolar concentration, can we see some dose-dependent killing? This is the same experiment, but just being done with lower concentration and on the transmitted founder virus-infected primary cells. As you can see here, no killing of the infected or uninfected cells. When you look on those 3BNC60 and 3BNC117, you see that there is better killing, and we see some dose-dependent killing when we start with 10 picomolar to 500 picomolar. Then we look at the other MicAbodies. We can see that PGT121 kills nicely as well, but 1074 just doesn't kill the HIV-infected cells. And that should not be surprising, since we are using bNAbs. Those bNAbs can kill vast amount of different strains, but they would not kill all the strains. So we discovered that 1074 is not a good candidate to kill this specific transmitted founder virus that we're using. And that brought us to think, what we need to do next? And we need to have more than one antibody, because if we use this 1074 against patient viruses, that they might be resistant to. We thought, okay, where can we have a greater breadth of killing than one antibody? And even what's going to happen when we have an escape mutant? So one is not enough, and we thought, can we multiplex? Can we have more than one antibody at the same time on the same CAR-T that covers more strains and might give us better killing efficiencies? So we did an experiment where in the same well, we had HIV-infected cells, and we also had B cells in the same well, and we used the HIV MicAbodies to kill the HIV cells, and the MicAbody that binds to B cell or CD19 or rituximab to kill the B cells. And we wanted to see, can we multiplex? Can we use at the same time 2 different cell types, MicAbodies, and manage to kill them both? As you can see here, the top is the killing of B cells, on the bottom is killing of HIV-infected cells. As you can see, the left 3 bars are the controls. What happens if you don't have CAR-T, or adding the CAR-T without the antibody, or the parental cells with the antibody? There is no killing of B cells or HIV-infected cells. But what happens when you just add to the ConvertibleCAR-T HIV MicAbodies. And as you can see on the, when you add the B cells MicAbody, you can see on the top it kills B cells in a nice way, and on the bottom it doesn't kill the HIV infected cells. If you add HIV MicAbody, you kill HIV infected cells, but you do not kill the B cell in the same well. But what happens if you add the B cell and the HIV MicAbodies together? You're getting nice killing of B cells and HIV infected cells in the same well, which shows us that now we can do multiplexing. We can use it at the same time. And what we were asking ourselves next is, this system is not the normal system. It's not just the convertible CAR-T or the conventional CAR-T binding to the effector cells. This is a three-body collision, which means that we have the effector, the target, and the antibody that goes between the target and the effector. And we ask ourselves, what is the time course of killing? And in order to do that, we collaborated with Dr. Nonvari, which did some time-lapse microscopy to track the kinetics of a single cell in a single cell level. And what we did, we took photos every 30 minutes, as you can see here in the video. The red cells are the convertible CAR-T. The green cells are the GFP positive or HIV infected cells. This is one of 20 fields of view that we took every 30 minutes for 48 hours. And what we wanted to see is that the GFP is being killed by the convertible CAR-T. When you take all the data and plot it on a graph, you can see that compared to the CAR-T by themselves with no MicAbody in blue, when you add the HIV mix, we used here all 4 different HIV MicAbodies, we get almost complete killing of the GFP positive cells in only 48 hours, which is something that was really nice to see, that when we combine more than one antibody, we can get better killing. And it takes some time. So since it's a 3-body collision, it doesn't happen right away. It takes between 10 to 15 hours to those 3 parts to come into contact and then kill the infected cells. And all I showed you until now were experiments that we did on healthy donors, that we infected them ex vivo with different HIV strains. But what I told you in the beginning is that our goal is to reduce the reservoir. So we need to see some samples from HIV-infected individuals and then try to see if we can kill those reservoir cells. So we ask ourselves, what can we get from HIV-infected individuals to do those experiments? Unfortunately, getting tonsils and spleen from HIV-infected individuals is not something that is easily obtained, but some HIV-infected individuals are generous enough to donate some of their blood. So this is the gold standard today in the field, using blood from HIV-infected individuals and do some experiment on that to kill the reservoir. What we did was lucky enough to get 6 leukapaks, 6 bloods from HIV-infected donors. We isolated those CD4 T cells where the reservoir is. We reactivate them for 3 days and then combine them with our convertible CAR-T. So 2 differences between the experiment before and now. First, we do not know which specific strain we have with any of those patients. So we use all 4 antibodies, all 4 MicAbodies against each of those reservoir. And another thing, since it's a wild-type virus, it doesn't have any GFP marker, we have to measure the reservoir in a different way. So after the 48 hours of incubation, we extracted the cell-associated RNA, and we measured by ddPCR the viral RNA. So this is what we were doing for those 6 patients, and we wanted to see, do we manage to kill the reservoir? And what we discovered, really for us to see, is that after only 48 hours of incubation, when you compare the reservoir size, as we can see here in green, with the CD8 with the highest amount of MicAbody mix, when you compare it to CAR-T, when you add 2 different concentrations of MicAbody mix, we get between 50 to 60% of killing of those HIV-infected cells or the inducible latent reservoir, which is good. This is not the best thing that you can do. 50% is not enough in order to have a functional cure. But if you remember, I told you that our convertible CAR-T we can maintain them in the body and then rejuvenate them over and over again. So if every time you get 50%, you can do multiple cycles and reduce substantially the latent reservoir. So I would like to summarize the result that I showed you today, and I hope that I convince you that the convertible CAR-T system is inert and it's safe to non-targeting cells. Highly selective to the target that you want. It basically can kill HIV-infected cells from blood, tonsils, and spleen. It can kill B cells if we want them, but really with high specificity. And when we looked at reactivated cells from HIV-infected individuals on ART, we could kill 50% of this reactivated reservoir. And what we think to do next is to use those convertible CAR-T in vivo with the MIC adapter rejuvenation. So we can do multiple rounds of latency reversing agents. So we can reactivate some of those HIV-infected cells and then kill those with those specific convertible CAR-T that we have. And we can do that in multiple rounds and get better results. And this is part of the thing that we're doing right now in mice. Some advantages that I want to share with you for the convertible CAR-T is that as opposed to normal CAR-T, you can multiplex and have more than one antibody. You increase the breadth. It's all the components are human, so you reduce the immunogenicity. You can control this system, which is one of the highest problem, or the biggest problem today in CAR-T, control your CAR-T. So we can either kill those cells with the MIC-A kill switch if those cells go out of hand, or rejuvenate those cells and make higher numbers of CAR-T cells in the body, and then have them kill those target cells over and over again. The important thing is that you don't need to continuously give new CAR-T to people. So you can give them only once, and then the physician can decide which antibody or which MicAbody is good for each patient, and you can exchange it, and it's pretty cheap to have those MicAbodies as opposed to have those CAR-T cells. That brings us to the next question, or the next problem that we have is, can we do that in HIV? So since autologous CAR-T cells are too expensive for giving it to everybody, it's around $500,000 right now to give it to cancer patients. So since we have ART, it seems reasonable to give it to people right now. But once we have something we call universal donors, those are cells that are not extracted from the patient, but are being produced in the lab, the price is going to be much lower, and then it might be feasible to get it to HIV-infected individuals. The LRAs, or the latency reversing agents that we use in our research, are not suitable for in vivo. They have high toxicity, so we urgently need safe and effective LRAs in order to get functional cure. We're working on that as well in our lab. One other thing is that, as I showed in the beginning, those reservoirs are not circulating in the blood. They are in sanctuary places like the lymphoid follicles. So you might need to target our CAR-T cells to those follicles, for instance, by expressing CCR5 on the surface, which would target them to the lymphoid follicle. And most, I would like to thank everybody that was part of this project, everybody at Gladstone, Warner, Andrea, Tom, and Roland, and Lior and Noam for time-lapse microscopy, the guys from UCSF that helped us obtaining the blood from patients, Steve Dix and Rebecca Hall, all our collaborators at Cyphos, that without them the project would not have worked. And of course, we would like to acknowledge the contribution that those HIV-infected individuals gave us by participating and giving their blood to our research. And I would like to thank you all for your attention. I'll be happy to take any questions.
Thank you, Eytan. Joy speaking. So I actually have a question regarding the technology. Yeah, okay. So I'm curious, how did you identify this alpha-1, alpha-2 domain and the MIC binding domain? Because there are two mutants and they have high affinity, and they don't bind to the wild type counterparts. So I wonder, how did you identify these mutants, and how you get this idea to combine this with the CAR-T and the antibody? Yeah, thank you.
Okay, so the mutation, the idea was pretty close by, since NKG2D and MICA are part of the killing mechanism of NK cells. So it's, for everybody that is using CAR-T, this is something that we're working on it a lot. So that make pretty basic receptor and ligand. And we, even in the beginning, we thought about using this natural NKG2D to kill HIV infected cells, well, just with the MicAbodies with no CAR-T. So it started in a different way. And those mutations that we're talking about in the NKG2D and in the part of the alpha-1, alpha-2 are basically were made by affinity maturation. You do some mutation and you see what mutations give you the best binding. This is how it was done.
Yeah, thank you. But if these, I wonder, these mutations you designed artificially, or do they exist in some patients?
They're artificially designed.
Okay, then there's another question about the safety or the immunogenicity of these mutants. Is that a concern? Because I know you show in the previous slide in mouse model that works when you treat the mouse multiple times, but is that a concern in human if that's a mutant?
Yeah, so it was checked. So basically, those are NKG2D with 1 or 2 mutants. So they're human proteins, and those specific mutants do not induce any toxicity or any immunogenicity. That's what it was found, and it's much safer since it's only bind to the mutant alpha-1, alpha-2, so it would not bind to the natural ligands in the body as we have others. So.
Yeah. Okay. And the second question I have, or the third question, it's also related to the safety. That's the CRS. We know CAR-T may induce CRS. So if this combined convertible CAR-T is infused to patients, will CRS be a concern?
Yeah, so this is a great question, because one of the best things that in this convertible CAR-T you have is that if you just introduce those CAR-T cells into the patient, it doesn't induce anything. It's an inert platform unless you have those MicAbodies. So I believe that if once we have the convertible CAR-T with the MicAbody, if you can dose it, you can have lower toxicities. I don't think you're going to have different toxicities if you have the same amount, but the great things about the convertible CAR-T that you can dose in the MicAbodies. So the cells by themselves are inert. They do not, we think that they would not have any toxicity in the body by themselves. Hope that answered the question.
Yes, yes, thank you. And I think to all the participants, so if you would like to ask a question, please type them into the question box in your Q&A panel. And we have a question, I think, from Nathan. The question is, can CCAR-T cells be used for further experiments? Or can it be done on strain specific, like strain A and C?
I don't really understand the question. Strain A and C of what? Of HIV, of other viruses?
Can I paste it to all the patients, to all the participants? Sorry.
So it can be strain specific, but we are going towards something that would be a general for all the strains. So those bNAbs that we're talking about are not strain specific. They can go between strains. There are different bNAbs today that can neutralize more than 200 different strains. So they have strain A, strain C, all the strains inside them. I hope it answers the question. We can go on a specific strain, but it would be, I would think, less efficient. It's better to have more than one antibody that have a breadth of different strain that you can attack. So I would go with all the strains instead of just going specifically on different strain. But of course, if one day this thing going to be that cheap that we can do a specific strain to a specific person, of course we can come in with a different MicAbodies that are specific to this person's viruses. Any other questions? Sorry.
Okay. I don't think just a moment we have a.
Okay.
I wonder, yeah, to all the participants, we still have questions, but it's all to me privately in the chat panel. Is that possible to type the questions to Q&A? Then we can see it. But I will read this question. What potential clinical hurdles, since now there are, sorry, since now there are both CAR-T cells and antibody involved in the treatment. So what potential clinical hurdles, since now there are both CAR-T cells and antibody involved in the treatment?
So one of the problems that we might have is that those MicAbodies that we're thinking that are binding pretty tight to our CAR-T cells might not be there, and we'll have to do more dosing of those MicAbodies, where in the conventional CAR-T, you just deliver one the CAR-T and that's it. In our system, you'll have to deliver the ConvertibleCAR-T in one time and the antibodies in a different time. But this is a hurdle, but it might be an advantage because if something happens, the toxicity might be lower because those antibodies are going to be degraded after a couple of days. So if there is any toxicity, we can stop giving those MicAbodies. If we can control those toxicities, we can give back those MicAbodies again and mix and match different MicAbodies that have lower toxicities and so on and so forth. That's something that I would see a hurdle to bring CAR-T cells. And when we talk about HIV, of course, it's the cost of this treatment. CAR-T cells are still very expensive. So that's why I would think that it's a problematic solution for right now for HIV-infected individuals, but in the future, the prices would go down and we'll be able to treat HIV-infected individuals as we treat cancer patients today. It's answering the question.
Yeah, thank you. And again, a question from Nathan. Is there any on-off switch to control or regulate the CAR-T cells in order to decrease cytokine storm or autoimmunity?
Yeah, so this is a great question, since in ConvertibleCAR-T, since it's something that you can send different signals to the CAR-T while you're in vivo, you have two parts. First of all, those antibodies that we're using, those MicAbodies, are going to be degraded after a couple of days. So if you have something of a toxicity, this toxicity is going to wind down over a couple of days, where in conventional CAR-T, once those CAR-T are inside, they're going to have those toxicities over and over again. And this is something that always going to happen. You're going to have lower toxicities in one hand, and for the question about kill switches, as I showed before, everything that you bind to those alpha-1, alpha-2 of MIC, you can deliver specifically to those ConvertibleCAR-T. And one of the things that we can do is send kill switches, which sometimes can be different complement proteins that we can bring to our ConvertibleCAR-T, and those ConvertibleCAR-T going to be eliminated. So this is our way to make a kill switch if we want. But I want to stress that convertible CAR-T by itself should be completely safe. It should not interact with any of the other cells and should not make any cytotoxicity unless MicAbodies are around. And the MicAbody should be MicAbodies that are specific to that specific target. If you do not have any target, you would not get any activation of those convertible CAR-T.
Thank you. I don't think we have further questions. Did you see any questions from your panel?
I don't see it on my end.
Okay, so then it looks like we've covered all our questions. So yeah, Eytan, is there anything else you wanted to cover before we wrap up?
No, thank you.
Okay.
Everything.
Yeah. Okay. Thank you, Eytan, and thank you, everyone. We appreciate you being here. Please visit Creative Biolabs website for updates, and if you have further questions, please email us at info@creativebiolabs.com. And thanks again for joining us today, and we will see you next time.
Thank you, Joy. Bye-bye.
Bye.